All right, hi, I'm Mark. I'm going to talk about orthology. I'm sure a lot of the groups got into orthology as we were talking about Syntony, and I'm going to talk more about orthology and how we determine it and how we use it at ViewPath. And one of those ways that we use it is in that syntenic view. So what is orthology? And I'll just give a brief introduction to what we think about it in terms of orthology. We're talking about two species, and like Plasmodium and Trypanosomes, and they may have each have an alpha tubulin, each have a beta tubulin, and actually the Trypanosoma brucei has another beta tubulin called beta 2 tubulin. How did they arise? Well, they arose from a common ancestor. That common ancestor had that alpha version and that beta version. And through a speciation, you ended up with the two species, and they each had an alpha tubulin and a beta tubulin. So those are considered orthologs. Orthologs are created through the process of speciation. They just maintain the alpha and the beta versions of tubulin. Then Trypanosoma goes on about its life, and it actually duplicates the beta version of the gene, and it creates a beta 2. And those are termed paralogs. When you have more than one copy of a gene, it's resulting from gene duplication. Those are called paralogs or Paralogus genes. Then the beta tubulin in plasmo is actually related to beta 2 tubulin in trypanosomes in a relationship called co-orthology, co-orthologs. That means that they're orthologs and resulting from gene duplication. So they're related by speciation and gene duplication. And some people ask, alpha and beta tubulins, like in Plasmodium, they're related, so shouldn't they be called paralogs as well? And the answer is yes, they are related. And they probably came from the same gene in a common ancestor way back when, but they have diverged so much. They became alpha tubulin and beta tubulin. They have totally different functions, and they have been maintained through the process of speciation. So they're diverged enough to now be considered different genes, not paralogs. So how do we use this information at ViewPath sites? So one place is on the gene page. You can see on the gene page, you can go to the Orthology and Syntony section, and you can see in there that a gene has a particular ortholog group, OG6148-264 for this particular gene, in Trypanosoma brucei, and all genes that are considered orthologs or paralogs of this gene also are put into this ortholog group. So you can click on this this link here to go to the OrthoMCL website, which is OrthoMCL is another website that we employ and use to create and display orthology relationships. So I can go to this group page at OrthoMCL. I should go back. I chose zeta tubulin, which is actually unique tubulin to this group of species. And actually, if you go to the group page at OrthoMCL, you can see all the species. There's 544 species there right now. And the number in the box shows how many genes within that species actually are in this ortholog group. And you'll see here's Trypanosome, and it has 2 genes in this group. And all euglenozoa actually contain genes in this group. But it's really interesting, as expected, zeta-tubulin is not found in any other species. This is fungal, chordates, bacteria, archaea. So OrthoMCL is the website that actually determines ortholog information, and it's another it's an algorithm that David Roos developed many years ago, and that we employ to determine the relationships, and connects all the genes on all our sites through Orthology.
So, sorry to break in, Mark, but just I do need to be accurate in correcting this. This was developed in my lab, but Li Li and Feng Chen really deserve all the credit.
Yeah, that's an important point. So it's that website. I just wanted to highlight one use there. I just showed you one. Here's another one. I look at that group, and I can look at the domain architecture of all the orthologs, and you can see they all share this tubulin domain, two copies of this domain. So it's really helpful for understanding the structure, the sequences of all of the orthologs of your gene of interest. So now going back to the gene page at ViewPath, I can go, this is what I showed you before, I can go further down in the same section, and I can look at Syntony, and you already talked about Syntony, so I won't spend too much time on it, but this is all determined from the same ortholog group. If you follow the gray shading down here, you can see the orthologs in different species, and that is determined at OrthoMCL, and that's displayed here. So then I can go further down on the gene page and see a little bit more cool stuff that Orthology is doing. So I have here a different gene. This is a gene page at Plasmo, and this gene is interesting because it has an unknown function. And so you maybe get that in your results and wonder, what is it doing? Well, we use Orthology to make predictions about its function. So it's in this ortholog group, and here's the number right here. And a lot of the genes within that group actually have this enzyme classification number 1.1.1.42, which means it's an isocitrate dehydrogenase. So it also likely is an isocitrate dehydrogenase. And we can further, because it has that EC number, it's been inferred to have that EC number, it's inferred to also participate in these metabolic pathways that are shown below on the gene page. So it might be involved in biosynthesis of antibiotics or in the citric acid cycle. So Orthology is really useful and is used throughout the website. So some orthology searches on ViewPath sites. So you might have a question, a specific question. Are there genes conserved in T. gondii, but they're not conserved in humans? Like maybe you're interested in drug targets and you don't want to find there's genes in humans that would react to this particular drug and be toxic. And the way to do this is use the Orthology phylogenetic profile search. So that search will bring you to this search page, and I'm going to look in these organisms in ME49 strain, and then I'm going to select the Orthology profile that I'm interested in. I click ME49 strain because I want to find genes in this group, but I don't want to find anything in mammals. I said humans, but I'm just going to say I don't want to see any orthologs in humans, in mammals at all. To get at my question, are there any genes conserved in T. gondii but not in humans? So when I do that search, this is the gene list that I get. There's actually a lot of genes, and this seemed like an interesting group of genes that are involved the cysteine proteases. They have an enzymatic function which could be druggable. And if I look at on OrthoMCL, I click on this link, it brings me over here. I can see all the species at OrthoMCL, and I can see where is this gene conserved, and it's just conserved in this group of Coccidia. Okay, so a last question, and we got to this question in my group, and it's all about an orthology transform. So say you're interested in C. tizeri, and you want to find out which genes are upregulated in C. tizeri. So we're interested in upregulation, so we're looking in transcriptomics, RNA-seq evidence, and we look at the experiments that are available on the website for RNA-seq. Unfortunately, there's no C. tizeri. So not to worry. We can choose another one that seems to be related to what we're interested in, which is oocysts. And I do a search for greater than 5-fold in oocysts, and I get 114 genes in C. parvum, which is a related species. Then what I can do is to add a step. And what I'm going to do is add a step to try to get the orthologs of those genes in C. tizeri. And you can add a step, transform into related records. Then it'll pop up what kind of record. I want orthologs. Okay, what organism do you want orthologs in? I want C. tizeri. And you can see that there are 133 genes in C. tizeri, which are orthologs of these genes that are upregulated in C. parvum. And if I look at these genes, they're pretty interesting. There's ABC transporters, et cetera. But one of the genes, I actually did not plan this, but I actually found that one of the genes was an oocyst wall protein, which seems relevant in C. tizeri and addresses my question. So we can use orthology, and we use that across the Viewpath sites, and it's very useful in understanding function in species that have data compared to species that don't have data. And this is the last search I wanted to tell you about, the paralog counts. Maybe I'm interested in Babesia, which genes have several paralogs, so may have represented a gene expansion event. And this paralog count is useful for doing that. And I can click on the organism, how many paralogs do I want to get? And I find 121 genes. And it's interesting that's part of 6 groups. So on average, these genes are in about 20, there's 20 per group. And you can see this SM-ORF set of genes, which are actually a well-characterized, not well, but it's a known gene expansion family in Babesia, and there's 41 SMORF genes in that species. So that's all I wanted to talk about.
There was a question, I don't know if from a user who's asking, and I think other people on the call can answer this as well. But if there's so many genes that you can infer their function based on orthology, then why not use this and annotate every hypothetical gene with its orthologous function, basically? And so, yeah. I don't know if you want to take a stab at this before, David?
Yes, we do that. So that is done in that EC inference thing that I was talking about here.
Right, but we don't take that and change the gene name then to say, okay, now this is probably a
so this is something that we've thought about as well. So we currently propagate these EC numbers to orthologs. The question maybe here is, should this be changed, this unknown function, should it be changed to isocitrate dehydrogenase? And that's something we're thinking of doing as well, is propagating the protein description and even the Go categories, all of that information to but the caveat is that it's just inferred. So maybe we'd have to have something in the description that says the inferred description.
Yeah, I
Let me
think
add
so.
I'm sorry if I said, Bettina, I think you were trying to say something?
Well, actually, I'm so sorry, but be careful with things like this.
Yes, exactly. So that's just what I was going to respond. So it's a very good question, and it's a question we all encounter. In fact, implicitly, you've all seen this in the Go enrichment. When you did your Go enrichment search, you were asked whether you wanted to look for Go term enrichment based solely on manually curated, highly validated Go terms, or those Go terms that have been inferred by similarity, based, for example, on PFAM domains, or in some cases on orthology. And in the query that we ran, we took advantage of that ability to infer function. For example, if we look in an ortholog group and find 100 genes in 100 different species, all of which are related to each other and none of which are related to anything else, and 40 of those genes have been annotated with a specific EC number, there is no realistic doubt that the other 10 should be and could reasonably be annotated with that EC number. But by the same token, there are also more distantly related genes where the question is a little more iffy. And exactly as Bettina has just suggested, the dangers of false positives are much greater than the dangers of false negatives. If you go and look at a gene and it is annotated as hypothetical, you realize that if you want to know more about that, you need to look into it in detail, into its orthologs, into its expression, into particular residues, into PFAM domains, and so on. But if you look at a gene and see that it's annotated as a tyrosine kinase, and you think, wait a minute, how can that be? I don't think that Trichomonas has any tyrosine kinases. You don't know who provided that annotation or why, so you're very reluctant to exclude it. Or worse still, maybe you just look at it and take it for granted that this is indeed a tyrosine kinase, which is completely wrong. So the dangers of false negative annotation and false positive annotation are much greater, and we've taken a very cautious role to not transferring that information. But this is exactly the reason for workshops like this. It's imperative that it's incumbent upon you and everyone working on a gene to know what the available resources are, to make your own interpretations and understand what the underlying evidence is.